So I will try to answer. The question whether it's safe or not safe to yield the mold gems. So can I ask for the next slide? Yeah, I think that it happened to all of us in our life that you go to the fridge, you just open your jam, and instead of a nice jam, you find something fuzzy and bitter. Which you definitely don't want to eat. So these are moths. Where the moths do they come from? So they are just part of our nature. They are everywhere. They are part of each ecosystem on the earth. Uh, but they play the although we don't want to see them on our food or on our fruit and vegetable, they play an important role in the ecosystem in biodegradation because they play the role in the recycling of the nutrients. We use them also in biotechnology and food industry for harvesting the pigments or producing some beverages and cheeses. And they are used also in the pharmacology for production of antibiotics, other various drugs and enzymes. On the other hand, as I said, we really don't like them when it comes to our food. They can also be spread and grow on the walls and thus destroy the property in our household. And some of them can also produce the toxins, which are called mycotoxins. These are the secondary products of some of the species, which can be hazardous to humans and animal health because they have a lot of mode of actions depending on their structure and physical chemical properties. So there are most of the mycotoxins are nephrotoxic, hepatotoxic. Some of them are also potential human carcinogens, and most of them causes immunosuppression, which can be also beneficial because in the very small amount they can be used as an immunosuppressive for people who have transplanted organs. Next slide, please. So coming back to the mycotoxins and to their producers. So what happens? So mycotoxins can be produced, or our food, our all the food we produce can be spoiled by the moths already in the pre-harvest period. So in case of apples, for example, they are already infested by the pathogens and other microorganisms on the orchards. But in this case, the toxinogenic fungi, the toxinogenic genera has to fight for their space in the ecosystem with also other non-toxinogenic microorganisms. Therefore, they don't have enough space for the further growth and spread. When it comes then, but they are already there in the spores or in small myceliums, which we don't see by eyes. But then when it comes to the post-harvest period under storage conditions, where there are not that many other microorganisms, they can grow and spread very fast if the conditions are favorable for them, and also produce mycotoxins. So most of the pathogens which can which can infect fruits and vegetables belongs to the genera of Alternaria, Penicillium, mm, and uh, also Cladosporium and Fusarium. So next slide, please. So this scheme is just for imagination that how many species can be found on fruit and vegetable. There are definitely far more. This is just the basic. And you can see that one mycotoxin can be produced by many species. And it is true also from the other ways around, that one species can produce a lot of mycotoxins, so the whole spectrum of mycotoxins. So, so this is just for you to see how uh, huge it can be in terms of the production of mycotoxins. So next slide, please. Then I would like to talk about the mycotoxins and food processing. So when the contamination comes from already at the pre-harvest period, mycotoxins are very stable, so they can survive all the processing and then ended up in the final product. Another source of contamination can be during the post-harvest storage, when the spores which are there already, or they contaminate again our stored products, can grow and produce more mycotoxins or different type of mycotoxins. Then there is really a high risk of contamination. In some of infections, like for example in Penicillium expansum, which causes external infection in most of the case, we can remove the moldy parts of the fruit, and then we then decrease the risk of the byproducts contamination. In most of the cases, the contamination is spread through the whole apple or the whole fruit, so we cannot just remove some parts, and then there is a high risk of the contamination of the final product. Next slide, please. So the most common mycotoxins found in fruits are those mentioned here, which are patchoulin, ochratoxin, citrin, aflatoxins, but mainly in the warmer areas, not in the mild areas like Europe, and alternaria toxins. As they can be harmful to human health, their maximum levels in certain foodstuff are regulated by the regulation 1881 from 2006. However, for the fruit jams, there is no regulation for such toxins. Next slide, please. So I had a look, what are the regulations or any recommendations from the food safety authorities on moldy food. So in the European countries, in general, from the European side, there is no official recommendation what to do when we have the moldy food. But for example, Swedish authority recommends discard moldy food in general. UK Food Standard Agency recommends discard obviously moldy food or rotten food to avoid to have the health risk. And United States, they are more specific, so they have the recommendation for specifically for jam and jellies, and they recommend to discard the whole jar when you find out that you have some mold in there. Next slide, please. Therefore, I was very surprised when I hear that the former Prime Minister said at one discussion about the food waste that it's fine if we just scrape all the mold from the surface of the jam and then eat it. That she does it the whole life and nothing happens to her. So I searched further and found out that also one mycologist from United States said for one BBC documentary that it should be really fine if the mold is spread only a little bit, just to scrape it off and a few centimeters beneath this mold, and then the jam is safe to eat. So next slide, please. Therefore, I was very happy that we had the chance to get some moldy samples, which was collected by Manfred Gessinger from the Austrian local producers, and we could do the analysis. So we could we could investigate what malt was in there, and also what was the mycotoxin pattern produced by this malt. So we had 75 mouldy samples, which varied of the food content, and also from the preservative which was used during the jam production. Next slide, please. So as I said, the goal of this study was to identify the fungi which was isolated from the jam surface. Then we also identified the metabolomic profile of this isolated fungi. We did it the way that we put them on the agar plate, and then we extracted let them grow, and then we extracted the fungi from the agar plate and investigated the mycotoxin profile which was produced there. And of course, we also determined the mycotoxins in the mold gem, and we were also curious whether the mycotoxin can penetrate deeper beneath the surface from the mycelium. So next slide, please. For that purpose, we used our method. So we sampled from 3 different spots. So we had from each gems 3 different samples. One sample was taken directly from the mycelium. One sample was taken 1 centimeter from this mycelium, but also from the surface. And the last sample, the third sample, was taken from beneath the mycelium, approximately 1 centimeter. Then we extracted it with acetonitrile water and formic acid, diluted the sample one by one to achieve the ratio of the 50% of organic solvent, and did the analysis using the liquid chromatography with tandem mass spectrometry. Next slide, please. So this is the description of our method. So currently we have the method we developed for analysis of mycotoxins and other biocompounds is able to determine more than 1,200 analytes in 40 minutes. So we have very broad spectrum of mycotoxins which we can detect with this method. And we also check whether the method is suitable for jams, because we didn't analyze that many jams from different fruits before. So we did also some method accuracy evaluations on spike metrics and corrected the final results on the recovery, so that we were sure that we really detected the correct value of the mycotoxins. Next slide, please. So, and now I'm coming to the results. So from all the samples, we mainly found Penicillium crustosum, Penicillium brevicompactum, and Aspergillus versicolor. These were the fungal species which were predominant in all the fruit samples. Next slide, please. Thank you. And I would like to show you the pattern which we detected from some examples of the fungus we identified out of these gem samples. So as I said, Penicillium crustosum was predominant in all the gem samples. So you can see the mycotoxin profile we found there. So from the fungal extract, we found the mycotoxins listed in the table, and except paspalin, all of them were found also in the gem. So, and you can see how they on the right side, you can see how they differ from the chemical structure, like sequopenol and roquefort-14 which are the most common mycotoxin produced by this species. Next slide, please. And here I have an example of one gem, which is the apricot gem. It was actually the sample which had the highest mycotoxin levels of all the gems we analyzed. And you can see that the highest levels of all mycotoxins were found as expected in the mycelium. But when we have a look 1 centimeter at the surface, 1 centimeter from the mycelium, we have very low levels of mycotoxins compared to the mycelium. And if we have a look into the depth of the gem beneath the mycelium, the levels also significantly decreased compared to the mycelium. So not all the mycotoxin penetrates from the mycelium, or it depends on their polarity, of course. So you can see that the cyclopenol penetrates slightly less than, slightly more than Roquefort 14, which is less polar than cyclopenol. Next slide, please. So another example is mixed fruit jam. We didn't know the fruit content. You can see that from the surface, the mycelium seems to be completely different, but it was the same species, Penicillium crustosum. And in this case, obviously, the mycelium wasn't that developed, that grown up, because the mycotoxin content was much lower. And you can see that if we have a look below the mycelium, 1 centimeter below this mycelium, the levels of all mycotoxins are almost at the detectable level, so almost nothing. If we didn't have that sensitive method, we wouldn't find them at all. So next slide, please. And now I'm coming to another example, which is the spectrum of mycotoxins produced by Aspergillus versicolor. So if we have a look on the metabolite profile from the Petri disk, we see that the spectrum of mycotoxin is much broader compared to gems. This is very known producer of sterigmatocystin, which was found in the fungal extract, but it wasn't found in the gem. But what we found in the gem, and you will see it also on the next slide, was two new metabolites, which are not that known from the literature, which is violacol 1 and violacol 2. Next slide, please. And violacol 1 was found at very high levels. This compound is not that known, so also the toxic effects are not fully investigated. And you can see that also 1 centimeter below the mycelium spot, we still have quite high concentration of this mycotoxin. So next slide, please. So I'm coming to the conclusion. So the predominant fungal species found in gems, in fruit gems, were Penicillium crustosum and Penicillium brevicompactum. The third one with the highest incidence was Aspergillus versicolor. The fungal metabolic profile basically matched the mycotoxin pattern in gem, and the highest levels, were found in the mycelium spot, much more, 20 times more, up to 20 times more than in the from the other parts of the gems. We can say that if you really scrap the visible mycelium and the few centimeters beneath of the mycelium, we can consider the gem as safe because we significantly reduce the health risk which can be caused by the consumption of mycotoxins. So with that, I would like to thank you, please next slide, for your attention. And I would like to thank also to my colleagues for their help, especially my friend Kessinger, who provided the samples, then Roman Labuda, who performed the fungal identification, and Marita Krpan, who did all the work and helped us also with the mycotoxin analysis. And of course, Rudi Krska and Karl Studiak, who helped with the financing of the whole project and also with the analysis of mycotoxin. With that, I would like to wish you a nice evening and thank you for your attention. Thank you very much. If there are any questions, I would be happy to answer them.
So, Mrs. Malachova, Thank you very much for your presentation. As we heard today, there are very different statements about the health value of processed fruit products. Scientifically precise research like the one you carried out is all the more important in order to clear up misunderstandings. Thank you very much.
Thank you.
Vielen Dank. Manfred, möchtest du noch etwas ergänzen?
I have to ask something. Thank you for the presentation. It's a wonderful overview about molds and jam and puree.
Thank you.
To be honest, it's a very surprising result you showed us, because even in school we learned that if we find any molds on food, we have to throw it away. And you say just put away only a few centimeters, and then you have no problems. My question is, because there were some a paper of Michael and Rudi about the speed of growing of molds in jam.
Mhm.
And it said that they grow really fast, about 1 centimeter per day or per week, I don't know. Do you think, is there a big effect of the matrix on the growth?
Yes, of course. The more liquid matrix you have, the easiest is that the fungus goes more into the depth of the jam. So it's easier to be spread, and also the water activity is very important, which is we have very high water content in all the jams, of course. So it really depends on the type of jam, how big we have the species, the pieces of the fruit in the jam. Of course, it's very important.
Is there one last question? Is it possible for a consumer to distinguish between the types of molds, the species? Are there some molds who grow faster and deeper in the matrix and others don't?
Yeah, it depends. It's a good question, because as we could see also on my two examples, that we have the same species. It was this Penicillium crustorium on two different type of jams. And on this apricot jam, we had very nice mycelium, grown-up mycelium. And on the other one, on the mixed jam, we had just a layer. So it was not like really visible nice mycelium, rounded mycelium. So, and it was the same fungus. So I think that laiks are not able to distinguish between the individual species. Sometimes even I have the problem, because I'm not the mycologist, I am the analytical chemist. So I really have to ask the people who works daily and who are very experienced in this area. So I think that normal consumers are not able to distinguish whether this malt is safe and this malt is not safe. So, and honestly, I would never eat moldy jams. Although we have these findings, and we see that really if you scrape, let's say, 2 centimeters or 3 centimeters from the surface, we still don't know enough. Maybe some mycotoxins are not there anymore, but some others can penetrate faster, but we just didn't detect them because we don't have standards for them. So if I see the mold there, I would just discard it and not eat it.
But you recommended just the opposite. Huh? That's
yeah.
I think it's
based on the
risky.
results, yeah, based on the results,
risky to
you really reduce the risk a lot. So for those who are, I know that many people are trying to reduce the waste, and from in all part of their life, like that they try to do the recycling economy, and they try to not discard the food which is not fully spoiled and so on. So me personally, I would not do it.
Ja, okay. Thank you.
Ja, vielen Dank. Also mir ist jetzt auch klar geworden, ich interpretiere Ihre Ergebnisse so, dass hoffentlich nicht so schlechte Zeiten kommen, aber wenn sie doch kommen sollten, dann könnten wir den Schimmel doch wegnehmen und dann den Rest essen. Ja? In diesem Sinne, ich glaube, der Reinhard hat eine ähnliche Frage noch gestellt. Do you have an idea how deep, far the mycotoxins could penetrate in jam? I think you answered it now. Maybe
yeah, and we will know
tell
more.
something.
We will know more about that because we did also further experiments like that. We spiked the jam, like the jam which was not contaminated, a healthy jam, with the species we isolated from these mouldy jams, and we let them grow for 2 weeks in the dark and under light at the different temperatures. And we really did the proper sampling with the knife that we really took 1 centimeter, 2 centimeters, 5 centimeters, 6 centimeters, and from the bottom. So we have also this experiment done, but it's not that evaluated. So the data evaluation is ongoing. So we will know exactly how deep they penetrate. And my guess is that each mycotoxin will penetrate at a different depth, depending on the structure. So we will know more soon.
So, vielen Dank. Also in einiger Zeit werden wir dann diese Frage auch von Ihnen beantwortet bekommen.
Yes.
Herzlichen Dank. Zum Schluss gebe ich.
Thank you very much.
Danke sehr. Ich